I'm back. So, in the last little video, talked about the sodium-potassium pump and the active transport that is being performed by the sodium-potassium pump creates a concentration gradient across most of our cell membranes, where there's lots of sodium outside the cell and very little sodium inside the cell. And that concentration gradient can be used to help us transport other things across the membrane. We sort of use one concentration gradient to push other substances across the cell membrane. And I want to wrap up talking about cell transport just by mentioning this important type of transport that we haven't talked about yet, and it's called cotransport. So we use concentration gradient for one substance as the driver for getting another substance into the cell. And you can pause the video here and read the notes, but this is one example of that. A glucose cotransporter that brings glucose into a cell along with sodium. And here's a more specific example of where that kind of cotransport takes place. This is showing a cell that is in our kidney tubules. And unfortunately, the excretory system was not chosen as one of our systems. I love the excretory system. You know, it's not a weird kind of love. It's an interesting system. That's the reason I love it. And so when we make urine, what we're doing essentially is filtering out the bad stuff from the liquid part of our blood. And the way it's accomplished is that we allow most of the liquid part of our blood to enter a system of tubules in our kidneys. And if the liquid stays in those tubules, it eventually becomes urine. But while it's moving through those tubules, the cells that are lining the tubules, like these cells in the diagram, are grabbing back all the good stuff that was in that blood plasma. It's called filtrate once it leaks into the kidney tubules. And so this stuff that starts off as more or less the liquid part of our blood is turned into urine as we recapture all the good stuff that was in that filtrate. And glucose cotransport is a key part of what's going on there. So in these cells that are lining these kidney tubules, glucose cotransporters are bringing back not only sodium ions, which are an important nutrient, but also glucose at the same time. The kidney tubules produce about a liter of filtrate per minute. And so you've probably guessed that we reabsorb most of that filtrate, or else we would be spending a lot of time in the bathroom. Okay, two other quick things, and then we're done with chapter 6. We're done with the transport part now. This is a slide that I'm just going to let you pause and read on your own. These are 3 different types of cell-to-cell connections, and these junctions serve a variety of different functions. And it's good to know that they exist. And when we talk about some of the tissues, specific tissues and organs and the functioning of those things as we get into the different body systems, you may be asked to hearken back to this brief introduction to cell junctions. All right, here's the last thing that we're going to talk about for chapter 6, the different types of signaling that takes place in the body. How is it that cells know what to do? How is it that cells know how and when to change their functioning. And the ways that signaling takes place in the body, the ways that we know, that our cells know how to respond appropriately to what's going on inside and outside of our bodies, is through these three different mechanisms, through paracrine, nerve, and endocrine communication. And I'll let you pause and take a look at these. Okay, so this is a brief overview of the different ways that chemical signal molecules are going to have their effects on different types of cells. And there are two basic categories of signaling molecules. Often we're talking about hormones here, but this could also be a neurotransmitter, and it could also be one of those paracrine regulators, one of those chemical signals that just acts locally. And the two different types are the water-soluble signaling molecules, which, because they are polar, they're water-soluble, they would not be able to pass through that non-polar interior in the lipid bilayer, in the plasma membrane. So those molecules cannot get into the cell, at least they can't do it very easily. And so they have their effects through second messenger systems. We'll talk about more of that in a second. And the second category would be the lipid-soluble regulatory molecules. And if they're lipid-soluble, they are nonpolar, and they have the ability to pass through that nonpolar interior in the lipid bilayer. So lipid-soluble molecules, nonpolar molecules, like the steroid hormones, they can pass through cell membranes, get right into the cytoplasm of the cell, and in fact sometimes pass right through the nuclear membrane and get right into the nucleus and have their effects there. Okay, so here we're going to look at the mechanism of a steroid hormone, how a steroid hormone can affect change in a cell. Remember, the steroids I just mentioned are lipid-soluble hormones. They can pass through the cell membrane, and I'm going to see if I can fire up this little animation.
Steroid hormones are not water-soluble. They travel in the blood attached to protein carriers. When steroid hormones arrive at their target cells, they dissociate from their protein carriers and pass through the plasma membrane of the cell. Some steroid hormones bind to specific receptor proteins in the cytoplasm and then move as a hormone receptor complex into the nucleus. Other steroids travel directly into the nucleus before encountering their receptor proteins.
So the receptor for the steroid hormone may be hanging out in the cytoplasm, or as they said, it may be hanging out in the nucleus. The steroid hormone by itself will not have any effect inside of that cell. That cell will only respond to the steroid hormone if the cell makes the specific receptor for that hormone.
Not shown. The hormone receptor protein, activated by binding to the hormone, is now able to bind to specific regions of the DNA. These DNA regions are known as the hormone response elements. The binding of the hormone receptor complex has a direct effect on the level of transcription at that site. Messenger RNA, mRNA, is produced, which then codes for the synthesis of specific proteins.
So the hormone receptor complex can get right into the nucleus, and in the diagram it shows you that the hormone receptor complex stimulates transcription. So a gene is activated, the messenger RNA is produced, that messenger RNA goes out to the cytoplasm, and a protein is made. Now the cell may be doing something different because it's produced a protein that it wasn't making before, or maybe it's making more of that protein than it was before. That hormone receptor complex can also bind to the DNA and inhibit transcription and translation. And so it could also be a signal to stop the production of a protein, which also might be an appropriate response given a particular situation. So that's what it looks like with a steroid hormone or other lipid-soluble signaling molecule that can get into the cell. Again, that molecule by itself in the cell won't do anything. It's got to bind to a receptor, and therefore the cell must make that receptor to respond to that particular signal, that hormone or that other signaling molecule. All right. The second type of pathway is when we have a polar molecule, a water-soluble molecule that is, because it's polar soluble, is not going to be able to pass through the non-polar lipid bilayer. And so these molecules can't get into the cell, so how do they affect the changes that they need to affect in their cells? Let's take a look at another animation.
When a signal molecule, such as epinephrine, binds to a cell surface receptor protein, it activates a G protein on the inside of the cell.
So epinephrine is a hormone. The old name is adrenaline.
The G protein then stimulates adenylyl cyclase to produce large amounts of cyclic AMP from ATP within the cell.
Okay, so the binding of the hormone, in this case the epinephrine, to its receptor caused a change in one of the other proteins, and it activated this enzyme adenylyl cyclase, which takes ATP and makes this other molecule called cyclic AMP, CAMP. And that is called the second messenger inside the cell. Because this receptor bound this hormone, a chain of events takes place, and now we have a second molecule, cyclic AMP, being released into the cytoplasm of the cell.
The cyclic AMP then binds to and activates a target protein, such as alpha kinase, which adds phosphates to specific proteins in the cell. The effect of this phosphorylation depends on the identity of the cell and the proteins that are phosphorylated.
So the cyclic AMP, the second messenger, is what's going to stimulate the cell response inside the cytoplasm. And it depends on the hormone, and it depends on the cell exactly what that response will be. But a very common effect of a second messenger like cyclic AMP is that some group of enzymes that were not activated will become active. Could also be switching them off, but a very common pathway would be to activate one or more groups of enzymes in the cell. And a very common target of this enzyme activation are groups of enzymes known as kinases. Kinases are enzymes that phosphorylate other molecules, and they do that by taking ATP and transferring that third phosphate, the terminal phosphate on ATP. They transfer that to another molecule, and that often activates or deactivates that other molecule. So it's again a somewhat convoluted pathway, but ultimately the signal at the surface of the cell, whatever that signaling molecule might be, a neurotransmitter, a hormone, that signal is going to initiate the activation of this enzyme that makes cyclic AMP or some other second messenger. And that second messenger is going to begin a cascade of events that leads to the cell doing something different.